Now that we know how to state our hypothesis, the next step of the scientific method is to design a controlled experiment. How do we design a controlled experiment? The first thing is, when we talk about a controlled experiment is an experiment in which only one variable is tested. In a controlled experiment, we test only one variable. For example, in this experiment, we are testing the effect of nitrogen on plant growth. So as you can see here in the control group, we are not adding any nitrogen, but in the experimental group, we are adding nitrogen. The difference between the control group and the experimental group is that in the control group, we don't add the variable that we are testing. In this case, the nitrogen. So the control group is treated the same way as the experimental group. The only difference between the control group and the experimental group is that in the control group, the variable that we are testing, we don't add it. But in the experimental group, we add the variable that we are testing so that we can see the effect of the variable that we are testing. The control group is a group that we use to compare the effect of the variable we are testing. So in this control group, we are using it to compare what will happen to the experimental group if we add nitrogen. The experimental group is a group that receives what we are testing. So in this case, it's only the experimental group that receives the nitrogen because the nitrogen is what we are testing. So that's the difference between the control group and the experimental group. The control group and the experimental group, they are treated the same, except the variable that we are testing. The variable that we are testing is given only to the experimental group. The control variables are the things that must remain constant in both the control group and the experimental group. So when you look at the control group and the experimental group, everything is the same, everything is constant. So these things which are the same or constant, they are called the control variables. So in this experiment, examples of control variables are the amount of water we add to the plant, the type of plant. We have the same type of plant, the same kind of plant, the size of the pot. The size of the pot in the control group and the experimental group, they are the same. They are identical. The amount of sunlight we give to the control group and the experimental group, they are also the same. So the only difference between the control group and the experimental group is the amount of nitrogen. Control group here doesn't receive the nitrogen, but the experimental group receives the nitrogen because we want to find out the effect of adding nitrogen to the plant. So these variables that we keep constant in the control group and the experimental group, they are called the control variables. So when we talk about the control variables, they are the things that we keep constant or the same in both the control group and the experimental group. Examples of control variables in this example, this experiment, are the amount of water we add. We add the same amount of water to the control group and also the experimental group. The type of plant, we have the same type of plant in the control group and also in the experimental group. The size of the pot, we have the same size of pot in control group and also the experimental group. We also have the same amount of sunlight in control group and experimental group. So in this example, the control variable are the things we keep the same in both the control group and the experimental group. They are the things that must remain constant. So whenever you are asked, how do you design an experiment to test the effect of nitrogen on plant growth? So when you are designing an experiment to test the effect of nitrogen on plant growth, you need to talk about a controlled experiment. That in a controlled experiment, only one variable is tested. And you need to mention that the variable you are testing in this experiment is the nitrogen that we are adding. You also need to talk about the control variables. What are some of the things that we keep the same or constant in this experiment? In the control group and the experimental group, the things that we keep constant or the same are the size of the pot. The same for control group, same for experimental group. You also need to talk about the same type of plant in control group and also the experimental group. The amount of water we add is the same for both. The same type of plant, also the same amount of sunlight. The only thing we change here is the amount of the nitrogen that we add to the experimental group. Then when you are designing an experiment, you should also talk about the step-by-step process that you take to do this experiment. So in this experiment, you can talk about obtain the same size of pot. You fill it with the same amount of soil in both the control group and the experimental group. You put in each pot the same height, the same identical plant. Then you give it the same amount of water in control group and experimental group. You give it the same amount of sunlight in both the control group and also the experimental group. Then you add the variable that you are testing to only the experimental group. So this is how we design a controlled experiment to test the effect of nitrogen on plant growth. Now let's take a look at this example. It's about hypothesis and control group. It says, a young scientist wondered if duckweed would grow faster in different kinds of water. The scientist set up 3 1-gallon containers filled with rainwater, fresh water, and salt water. So the scientist is using 3 different kinds of water: rainwater, fresh water, and also salt water. 4 individual duckweed plants were placed in each container and placed in a 21 degrees Celsius room temperature where all plants received the same type and amount of light. So this is designing an experiment. These are the control variables, the things that we are keeping the same in this experiment. The scientists counted the number of duckweed plants in each of the containers on 3 different days: day 4, day 10, and also day 14. So now let's look at the questions that follow. State one possible hypothesis for this experiment. So based on the experimental information, experimental setup, designing the experiment information that we've been given, it's asking us state one possible hypothesis for this experiment. So you can say that duckweed plants grow fastest in fresh water. So you can pick one of the different kinds of water. Then you say duckweed plants grow fastest in fresh water. So this is a hypothesis. This is a statement that we can test to see whether duckweed plants grows fastest in fresh water. So this is a hypothesis. The next question is identify 3 conditions other than the size of the container and the temperature of the room that should be held constant in this experiment. So this question is asking us control variables. What are some of the things we keep constant or the same in both the control variable, in both the control group and also the experimental group? So in this experiment, examples of control variables are, remember in this experiment we are testing which kind of water will duckweed plants grow in fastest. So you are going to keep everything the same. So the things you keep the same are the amount of sunlight you give to each gallon or each pot of water. The amount of water should also be the same. The same type of duckweed, we are using the same type of plant. So these are some of the conditions that must be kept constant or the same throughout the experiment. The only difference in this experiment is the kind of water or the type of water, because we are testing which kind or which type of water will duckweed grow in fastest. So that is what you are testing. Which kind of water will duckweed grow in fastest? And our hypothesis is duckweed plants grow fastest in fresh water. And it's because of the amount of nutrient or the required nutrient in fresh water. And we can conduct a controlled experiment to test if this hypothesis is true. The next thing we are going to look at is dependent and independent variable. Dependent and independent variable. We are still looking at the effect of adding nitrogen to plants. So when we look at the effect, this experimental setup, we see the same pot, the same type of pot. We see different amount of nitrogen that we are adding. So when we talk about dependent variable is what we measure. Dependent variable is what we measure or what we observe. In this experiment, what we are measuring or what we are observing is the height of the plant. So the first one, we see 2 centimeters. That's what we are measuring. The next one, the next plant is 8 centimeters. That's what we observe or we measure when we add the different amount of nitrogen fertilizer. The next one is 4 centimeters. This is what we measure. So what we measure or observe is the dependent variable. Like in this experiment, the height of the plant is a dependent variable. So whenever you are asked, identify the dependent variable in this experiment, you have to say that the dependent variable in this experiment is the height of the plant, because that's what you measure. You don't have to give the definition of dependent variable. It's asking you what is the dependent variable in this particular experiment. It's not asking for the definition of dependent variable. It's asking you to identify what the dependent variable is in this experiment. And in this particular experiment, the height of the plant is the dependent variable because that's what we measure or observe. The next one is the independent variable. When we talk about the independent variable is what we change or manipulate. The independent variable is what we change or manipulate. So in this experiment, the independent variable is the amount of nitrogen we add. As experimenters, the independent variable is what we can change or manipulate. So here we are adding 1 milliliters of nitrogen to the first plant. The next plant we are adding 10 milliliters of nitrogen. And the last plant we are adding 50 milliliters of nitrogen. So if you add different amount of nitrogen to the plant, how would that affect the plant, the height of the plant, or how tall or how big the plant will grow? So the independent variable is what we change as experimenters. And when you change the independent variable or when you manipulate the independent variable, it's going to have an effect on how tall the plant will grow or how big the plant will grow. And that is a dependent variable. What we are changing is the independent variable. So this means that if you add the right amount of nitrogen fertilizer to this plant, that means the plant will grow taller. If you add 1 milliliters, the plant will grow only 2 centimeters. If you add 10 milliliters of nitrogen, the plant will grow 8 centimeters, which is the tallest or the biggest amongst all the amounts of nitrogen we added. If you add 50 milliliters, the plant grew only 4 centimeters because having too much nitrogen can actually even kill the plant. So the independent variable is what we change. The independent variable is what we manipulate. So in this experiment, when you are asked to identify the independent variable, you have to say the independent variable is the amount of nitrogen we add to the plant, because the amount of nitrogen is what we change, is what we manipulate. Here in this experiment, we can choose to add 1 milliliters of nitrogen to the first plant. The next plant we can add 10 milliliters. The next plant we can even add 1,000 milliliters or 800 milliliters to the plant. So what we are changing, what we are manipulating is the independent variable. What we are measuring, what we are observing is a dependent variable. So that's the difference between dependent variable and independent variable. Dependent variable is what we measure or observe, like the height of the plant in this experiment. The independent variable is what we change or manipulate, like the amount of nitrogen we add